Okay, it looks like it's going live now. All right, so hello everyone. Good morning, good afternoon, good evening, wherever you are. Welcome to this week's Spine seminar. Today I'm happy to be a hostess. Looks like it's going live now. Whoops. I got the echo. All right, so hello everyone. Good morning. Apologize for that. As always, technical issues. But so I'm happy to be your host today for the Frontiers in Social Evolution seminar. My name is Lauren Hayes. I'm a professor at the University of Tennessee at Chattanooga in the United States, and I study mammalian social systems with a focus on a rodent in Chile. It's really my pleasure to be host today. And before I introduce today's speaker, I want to just mention a couple of things. First off, for those of you who are new to the Find, the seminar is about 45 minutes, followed up by up to an hour of question and answer. And we really encourage people from the students to get engaged and have conversations with the speaker. It's a great opportunity to make connections. Next week, we have Eugenia Natalie. She's going to be talking about multiple paternity. Her title of her talk is Multiple Paternity as a Measure of an Ineffective Male Reproductive Strategy, the case of free-ranging, unowned urban cats. And you may actually see one behind my house as they wander through my yard. But that's for our next week's speaker. This week, I'm happy to be host of Jenn Moss. Jenn is what I call a rising star in the U.S. She's an assistant professor, recently hired at the Virginia Tech University. Came highly recommended from some folks in the amphibian reptile world. And I'm really happy to be hosting her today. Just a little background on her work. As I said, she's a rising star in the field of behavioral ecology. Assistant professor, Virginia Tech, and she's had considerable postdoc experience. She has had postdocs at the University of Illinois with Eva Fisher, who I believe is a regular here. Postdoc at the University of Georgia. She did a postdoc in Tasmania, Australia. And prior to that, she earned her PhD at Mississippi State University, where she studied rock iguanas. And then her undergrad is at Hood College, which I believe is in Maryland in the United States, where she did an honors thesis on rays, so cartilaginous fish in the Chesapeake Bay. So she's studied a diversity of organisms. Jenn's primary research focus is on sexual and parental behaviors in various species, particularly in amphibians and reptiles. And she describes the two themes of her current work being looking at sex differences in behavior, how that shapes demographic and evolutionary responses to climate change. And she thinks about how animal parental behaviors and mechanisms inform evolutionary origins and diversification of parental care. So taking a comparative approach as well. In terms of productivity, she's done a really nice job publishing roughly 20 or so papers and listing Google Scholar in some really good journals recently in, I think, Proc B. There's an AmNat paper in there somewhere, Evolution, and the Behavior Journal. So she's doing really nice work in getting it out there. And I'm really excited to hear her talk today. For those of us who study mammals, this is a nice change. We're going to hear some more about other taxa. And just, for her talk today, the title of her talk is Unpacking Thermal Effects on Social Behavior through integrative approaches. And with that, I'm going to stop the share and turn it over to you, Jenn. And thank you for being here with us today.
Thank you so much, Lauren. Let me go ahead and try and share again. Make sure you can see my slides. All right. So, all right. Yes, thank you, Lauren, so much for that intro. I'm really honored and excited to be here. I think this is a fabulous seminar series. As Lauren described, my research foci over the last several years have jumped around quite a bit. I started out, actually, Lauren went back pretty far. So I actually started out working on rays before moving to lizards, which typically people think of as non-social, and then moved on to subsocial insects, and most recently with Ava, the neotropical poison frogs. And so when Lauren invited me to give this talk, I did struggle to decide what I was going to talk about and what would be broadly interesting to folks. Ultimately, what I decided to highlight is what's become a recurring theme in my research, which is looking at this intersection between climate change and social behavior, something that I think is really topical right now. And thinking about these ideas has shaped a lot of my perspectives currently on evolutionary questions, and in large part inspired the research program that I'm developing here at Virginia Tech in my new position. And so I hope people find it interesting, and I hope that it stimulates some conversation. All right, so as I'm sure no one in this audience needs convincing of, how organisms, individuals interact with each other is really foundational, not only to the ecological stability of populations, but also to shaping the mating and the social systems by which species organize themselves. And so for those of us who are interested in understanding how anthropogenic environmental changes are affecting wild organisms, we really need to recognize that those impacts are being filtered through a social environment, and that social interactions have the potential to really radically alter the relationship between organisms and their abiotic environment. Specifically, if we think about climate, scientists over the years have uncovered a wealth of evidence across a wide range of taxa supporting this idea that, at least historically, climate has been this really key ecological driver of evolutionary transitions and social traits. Thinking about organization of insect societies, social grouping in things like rodents and lizards, parental care and allocare in amphibians and birds. But when we really dig into these patterns, not unexpectedly, what we find is a ton of complexity, not only in terms of the specific climatic variables implicated and the direction of those effects from one lineage to the next, as new behaviors are formed and canalized out of existing structures. But what I think we're learning is that these phylogenetic signals provide a really nice guide for how things have happened in the past, but climate change is sort of its own beast. And the processes that were important historically for a species are not necessarily the same species that are, or the same processes that are unfolding today. And for that, we really need a deeper mechanistic understanding of how individual plasticity and socially relevant but climatically sensitive behaviors is playing out within existing social structures. And to take that one step further, we really need to understand how resulting social dynamics then shape selection on traits implicated in climate adaptation. So in an effort to get more at the nuts and bolts of this, I wrote a review back in 2021 with Jeff Weil, who was my postdoc advisor at the time. In it, we explored the different pathways through which one specific abiotic variable, being temperature, mediates social interactions through effects on individual behavior. So all organisms, be they endotherms or ectotherms, have a preferred range of temperatures for activity, meaning at the most fundamental level, the thermal environment determines when and where individuals are active or available for other individuals to interact with. Of course, we can think about this at the level of the individual organism. We can also scale up and think about this at the level of the social environment with which an individual is interacting. So as the thermal landscape shifts, the distribution, density, and demographics of the individuals encountered are going to shift along with it. All this to say that if activity depends on temperature, then so do the opportunities for social interaction. We can also extend this line of questioning to ask, what are individuals doing when they are active? Are they looking for mates? Are they engaged in competition? As thermoregulatory opportunities are shifting in their timing and intensity, individuals might have a harder, easier time engaging in certain behaviors, or may need to adjust how they're trading off between behaviors. And so in that sense, temperature also has this potential to shape the nature of social interactions. And then finally, what are the costs and benefits that individuals are reaping from interacting with others, as changes in temperature are imparting synergistic effects on things like resource acquisition and body condition, predation risk, thereby altering, we can think of as altering the context for social interactions. And just as an aside, here I'm using temperature as my example, but again, because so much of an individual's fitness depends on these social interactions, and really what this framework is getting at is how these indirect channels are filtering environmental variation. We could easily replace temperature here with a different variable and work out similar pathways. Underlying all of that complexity and interacting pathways is yet another level of complexity, which is the fact that wild populations are, of course, not homogeneous. They're amalgamations of age classes and sexes and morphotypes, each with distinct thermal sensitivities. And until recently, intraspecific variation in thermal responsiveness was really regarded as not that important relative to other factors for forecasting population responses to climate change. But what we're seeing is new and improved models accumulate is that that's really not the case, that accounting for individual variation can really radically alter our projection of how populations will fare under changing climates. And when the response we're trying to understand is social behavior, it's obviously quite critical to take stock of potential differences in the thermal responses of our participants. And in fact, it's even been suggested recently that so highly social species may see amplified effects of climate change, as this greater interdependence among individuals reinforces this kind of weak link ripple effect through the group. And so as changing environments are bringing out different phenotypes in our population, of course, we need to be thinking about the effects on individual fitness, but we also need to be thinking about reciprocal effects on interacting individuals. And it's this holistic approach that I think is going to be really important to determining who in the population might be disproportionately driving or even acting as a constraint on adaptation to new environments. One category of social interactions where I think this point is particularly cogent is interactions between the sexes. For species that reproduce sexually, these interactions are obviously imperative to population growth and species persistence. But more than that, and of particular interest to this audience, the mating system, so the rule book, if you will, for how individuals are successfully acquiring mates and investing in offspring, really shapes the evolution of so many other aspects of a species' biology, including how it organizes itself socially and how social bonds are persisting over time. And one of the things that I think makes interactions between the sexes so interesting in a climate change sense is that males and females are these physiologically and behaviorally distinct units of the population. And therefore, we have every reason to expect that they may show distinct responses to changing environments, which is especially true when it comes to interactions over reproduction, where not only are males and females at their most physiologically distinct, but the interests of the sexes also diverge during this time. And so using that framework of mechanistic pathways I outlined in my review, we might predict that a shift in temperature is going to be met with sex-specific effects on physiology, which could impact opportunities for interaction. There could also be sex-specific behavioral decisions about how energy is allocated to different kinds of interactions, or all of the above. And of course, because reproduction is a two-way street, responses of one sex have the potential to facilitate or constrain outcomes for the other sex. And what's interesting is that despite having a lot of really good reasons to anticipate that males and females might differ in their response to temperature, empirical evidence for these patterns in nature is really surprisingly limited. Traditionally, our approach to the field of thermal ecology has been either to ignore sex completely, so not report it, or to control for it by measuring responses only in a single sex. And where that leaves us is in really desperate need of more hypothesis-driven tests to understand how these effects could play out under different environmental conditions. So for today's talk, I'm going to be exploring these ideas through the lens of 2 case studies where I try to focus specifically on quantifying sex-specific thermal responses and then linking those to male-female interactions and their outcomes. And then with my last 5 minutes at the end, I'll wrap up with some future directions. So where I think are the most important places to focus empirical research effort to further understand these patterns, and how I envision that we could be leveraging those insights in a broader framework to understand how social interactions could be influencing climate adaptation. Okay, so digging into the first case study, I have 2 major questions I want to address. First, do opportunities for male-female interactions vary as a function of temperature? And second, does individual variation in thermal plasticity actually drive variation in mating success and by extension mediate the sexual selection that's happening in the population? So what classical theory tells us about this is that an animal's preferred temperature for activity, its thermal tolerance range, is a central pacemaker for broader demographic and life history patterns due to the simple fact that animals that are active are taking in energy, establishing and defending territories, and encountering potential mates at higher rates than individuals that are inactive. And so as the number of hours suitable for activity increases as a function of the thermal environment, we should see concomitant increases in energy allocation to reproduction and overall increased reproductive success. Specifically with regards to mating behavior, we have a lot of empirical evidence pointing to the fact that thermoregulatory behavior impacts mating success. So for example, in many species, the thermoregulatory opportunities conferred by a male's chosen territory or a signaling location directly impact ability to signal to potential mates and thereby attractiveness to potential mates. Whereas in other cases, thermoregulatory behavior can affect mating success through these interesting indirect avenues where potential mates are driven to aggregate in territories of specific individuals, and that confers disproportionate advantages. And so over short periods, we see temperature acting as really quite a powerful mediator of variation in mating success via ultimately this quite simple and universal mechanism, which is behavioral thermoregulation. But of course, it's a little bit more complicated than that because we know that the temperature animals prefer, or at least the temperatures they can stand to be active at, are in reality highly flexible. As environments change and thermal opportunity windows expand and contract, an animal's physiology is going to acclimate to make the most of the environment it's in. And of course, these changes in physiology can support patterns of activity outside of what's typically considered optimal. Bringing it back to sex, something that I think is really interesting to consider is that the degree of plasticity or of thermal acclimation could differ between the sexes. So if we consider most sexually reproducing species, the way males maximize reproductive output is by maximizing mating opportunities, whereas females typically have a more conservative strategy based on maximizing energetic intake and minimizing risk. And so what we see in some of these systems is that males will push themselves to thermal extremes beyond what they would normally tolerate if it means getting the chance to mate. Male fiddler crabs are a really fantastic example of this. So they court females only on these exposed beaches away from the water's edge that provide really optimal habitat for building nests, but that experience much higher temperatures than what the crabs would normally tolerate. And because females typically don't occupy these habitats at nearly the rate of males, what this creates is an opportunity for selection on thermal tolerance limits to operate in a sex-specific manner. And when we're thinking about climate adaptation, that sets the stage for some really interesting, I think, evolutionary dynamics. Another really key consideration is that the dynamics of sexual selection are going to fluctuate over time. Because sexual selection is frequency dependent, so it's contingent on operational sex ratios, or the number of receptive females there are to every one male, it's not enough to study the thermal responses of males in isolation. We also need to understand how shifts in thermal opportunity windows are going to alter the distribution and receptivity of females, and how activity patterns over time ultimately drive variance in mating success. While this is really obvious to say, surprisingly few studies have actually experimentally quantified the relationship between thermal plasticity, activity time, and mating performance in a population in both sexes simultaneously. And so this was a gap I wanted to attempt to fill with my first postdoc, which was work I did in Dr. Jeff Weil's lab with the help of an exceptional honors student, Zach Borthwick. Jeff's lab works on this really incredibly charismatic lizard called white skink, which is a family living skink, meaning that males and females like to co-defend territories. And this often allows offspring to delay dispersal and hang out with mom and dad for their first couple years of life. And so this is truly an example of where patterns of mating actually form the scaffold for the further stabilization and elaboration of social structures. And so when we're thinking about climate change, we have to remember that this is happening on a global scale, and the direction of effects are going to vary by location. So typically when people think of Australia, they're thinking about these very hot and arid environments. My work actually took place in Tasmania, which is this island off of the southeasternmost tip of the continent, and it experiences a highly temperate climate. And so if you're a lizard and your breeding season is in the austral spring, we might actually expect rising temperatures are going to alleviate constraints on activity. So because our goal was to understand how individual variation in activity translates into mating success across a season under differing thermal regimes, we knew that this was going to be an experiment of some scale. And so what we did was we actually created 8 replicate mini populations of lizards out in these massive outdoor enclosures called mesocosms. I think the really cool thing about the mesocosm approach is that it allows us to simulate different environments while maintaining natural variation in weather patterns over the course of a breeding season, such that we're really capturing realistic patterns of behavioral thermoregulation over time. So we had these 2 treatments that we created, unrestricted full sun and restricted sun, which is where we essentially draped this shade cloth over top of half of our enclosures. And if you're wondering what this treatment represents in the real world, we found that the average temperature differential between a shaded and unshaded enclosure was about 3 degrees Celsius, which is realistic in the context of future climate change, but it also happens to capture quite accurately the year-to-year temperature variation that's been recorded at our site over the last 40 years, where spring 2019, the year that we did this, the season that we did this experiment, was among the warmest on record. So we can kind of think of our treatments as corresponding to a relatively cool spring and what will be an increasingly normal warm spring for this site. All right, so here we can look at our temperature trends over the course of the season in the upper left corner here. You'll notice that we have consistent differences in temperature between our treatments. But more than that, we have that seasonal variation over the course of the season that I was talking about within thermal treatments. So I'm about to play some animations that will depict how lizards in the enclosures responded to that temperature variation in each of the 2 thermal treatments. And these are created from real activity paths reconstructed from individual positional locations recorded throughout the survey period. So here we're starting out, individuals are moving around a bit slow at the start of the season. Now watch as we hit this peak temperature around mid-October. Okay, yeah, so there was that explosion of activity there in both of the treatments. So there are definitely effects of temperature that we can see with the naked eye. But there's also a lot to unpack looking at these between treatment, seasonal timing, and sex. So we're going to break that down. In an effort to take all of that crazy variation and dial it into a number we can actually quantify, I extracted a bunch of different metrics from our observational data, so emergence probability, rate of movement, and I ran those through a PCA to give me a single variable reduced activity score for each individual on each day of the experiment. And then to analyze how activity levels respond to mean temperature in the different environments, I use this tool called the reaction norm, which is simply a mathematical description of how a given trait changes across a gradient of temperatures. And so here I've run actually a multiple regression. And so each of these lines represents a individual thermal response. And so here in our shaded enclosure, you'll notice that all of these reaction norms have fairly steep slopes, meaning that temperature in this environment is a really strong determinant of activity level. Now let's look at the same relationship in the sun enclosures, so the unrestricted sun enclosures. One thing I want to point out here is that the temperature scale on the x-axis has shifted to now include much warmer values, meaning individuals are now experiencing a wider range of temperatures, and yet the overall variation in activity is reduced across that range. So in reaction norm speak, we would say that these individuals show elevated intercepts, but reduced slopes compared to individuals in the shaded environment. So all this to say there's plasticity. If you take lizards from the same population and acclimate them to different seasonal thermal regimes, they're going to adjust their behavior to maximize activity across the range of temperatures they're most likely to experience that year. And in fact, if we were to compare the overall mean activity levels of lizards between the two treatments across the season, we would find that they are equivocal. So now let's re-examine these treatment differences, taking into account the sex of the individual. What we find is that it's really males who are the primary drivers of the thermal acclimation patterns. This is reflected in males having significantly steeper slopes than females in the shaded treatment, and significantly higher intercepts in the sun enclosures. So females on the whole actually don't appear to exhibit a very strong thermal acclimation response, at least in terms of their activity levels. Which brings us to the next question, which is, if the degree of acclimation differs between the sexes, then should we also expect to see differences in the degree of sex-specific selection for that response? So first, looking at our males, again, we predict that the majority of a male's reproductive fitness is going to be tied up in his mating success. We can confirm that with our data. If we reconstruct parentage of offspring using molecular fingerprinting, we find that the number of females that males successfully mate with accounts for the majority of variance in reproductive success. But what's more important for our questions is that we find, yes, activity is a really key driver of this. So in both environments, males who are able to maintain higher activity levels than conspecifics through their treatment-specific acclimation strategy sire the most offspring in their respective enclosures. By contrast, when we look at females whose fitness is largely tied to fecundity rather than mating rates, we find activity has no particular effect on mating rates. So presumably females don't need to be up and moving about to successfully secure mates. Instead, what we find is that females who are less active are simply less likely to reproduce at all, but only in the shaded enclosures. In sun, female activity has no effect on reproductive probability. So to recap here, sexual selection on male activity occurs in both environments. Fecundity selection on female activity occurs only in this more thermally restrictive environment. And so at this point, you might be asking, if females have less capacity for thermal acclimation than males, but suffer this profound fitness cost in certain environments in the form of lost reproductive opportunities, then shouldn't we expect environmentally dependent feedbacks on males, given that sexual selection is frequency dependent? So I thought this was a really interesting question. And we can actually get at this by calculating the intensity of sexual selection and the intensity of fecundity selection from environment-specific means and variances in reproductive output. And what we find when we do this is that even though sexual selection on males is acting quite strongly in both environments, it's actually lessened in the shaded environment because the reduced number of offspring available to sire enforces an upper limit on the variance in male reproductive success. And simultaneously, fecundity selection on females is much stronger in the shaded environment. And so when we solve for this equation, we get what we get is a larger ratio or a greater contribution of sexual selection on males relative to fecundity selection on females as thermal constraints on activity relax, which means that increasingly warm temperatures, at least in this population, in this location, could be permitting sexual selection to play a larger relative role in thermal adaptation, which I think is super exciting. And just as a fun follow-up, after I ran my experiment during the breeding season that year, one of the lab's PhD students, Alex Buffet-Halley, went out and ran another experiment where she introduced families with offspring to the thermal treatments during the austral summer, so a time when it gets really hot in Tasmania, and found that in this case, warmer temperatures tended to restrict activity, and that in turn limits the emergence of social networks among skinks. And so feeding into this narrative that effects of climate change are really complex and multifaceted, we see that in this population, warming during the spring could alleviate a lot of constraints and allow for the production of more offspring and heightened activity driven by sexual selection on males. Whereas in the summer, we're going to see reduced activity and weakening of social networks as interactions become less frequent. And all of this together could mean changes in the social and family structure of these lizards over time, which is really starting to get now to the meat of what we're trying to capture with these large climate simulation experiments. All right, so I now want to transition to my next case study, which is moving now to a topic that tends to be a bit more at the forefront of people's minds when they think about social behavior, and that's parental care. This is another realm where I think interactions between the sexes are very important and where the diverging interests of the sexes have the potential to influence climate change responses in some really interesting ways. And so leveraging this case study, I really want to delve more into how temperature modulates the nature and the context of interactions between the sexes, more so than just the opportunities. And I'm going to approach this by asking whether effects of thermal stress on the costs and benefits of care are associated with sex-specific changes in behavior. So the inspiration behind this project originated with what's known as the hostile environments hypothesis, which basically says that the joint rearing of young, so biparental or alloparental care, has pronounced benefits in harsh and unpredictable environments, and that these conditional benefits might compensate for the sexual and family conflict that would otherwise be dominating dynamics. So we can take zebra finches as an example, where parents invest less when they're caring with a partner because there's this disagreement over who should be the primary caregiver. And because under most conditions, a single parent is enough, offspring don't need two parents, this tug and pull is allowed to persist. On the other hand, when environmental conditions worsen and impose steep fitness costs on parents and offspring, such that a single parent might no longer be sufficient, Wilson and Emlyn would argue that biparental pairs or alloparental families should have a leg up because cumulatively they're capable of providing more care and may even help alleviate some cost to each other, depending on how the individual participants in that arrangement are responding behaviorally. Of course, empirical evidence for this is very mixed. Almost everything we know comes from birds, in which biparental care is very widespread, and the extent to which certain ecological factors may or may not be responsible for driving parental coordination can be really difficult to tease apart. because birds also have really high rates of social monogamy. So there can be a lot of confounding variables where an individual's investment in parental care is not just impacting their reproductive fitness, but also their social standing. And so to try and say something more generalizable about how environment is selecting for a number of caregivers, we really need to study this in systems where cooperation is restricted to the reproductive event. And so enter Necrophorus orbicollis, the sexton burying beetle. This is a species of carrion beetle that utilizes small vertebrate carcasses as breeding resources. As you might imagine, most of these species do best at mid to high latitudes where temperatures are cooler and things are taking longer to decompose. But orbicollis, which just happens to be a biparental species, is one of the few that's managed to break into these lower latitudes of the southeastern US, where temperatures during the breeding seasons get really warm. We know that temperature is really limiting to reproductive success in this species based on a mounting number of studies, both observational and experimental, which clearly show success starting to decline at temperatures above 20 degrees. But what's interesting is that despite those fitness costs, we don't see any compensatory shifts in phenology of populations inhabiting these warmer latitudes. Midsummer is still the peak breeding season for this species, even in its southern range limit. And so this was a puzzle that I wanted to set out to solve when I joined Dr. Alan Moore's lab at UGA, was this question of, if beetles are not avoiding these high temperatures, are they finding other ways to cope with the challenge? Before I go any further, I just wanted to give a quick and dirty breakdown of what reproduction actually looks like in burying beetles, in case anyone is unfamiliar. As I said, these guys breed on small vertebrate carcasses, which are these highly ephemeral resources for which they face considerable competition. And so when the species is biparental, males and females will work together to bury it so it's not discovered by other decomposers. And there the carcass is molded into this nice brood ball, which is then colonized by larvae hatching out of eggs nearby. And then comes what I think is the most fascinating part of this beetle's biology, which is quite unique to this genus. And that's that parents actually directly provision their young by regurgitating pre-digested carrion into their mouths. This is also the most energetically taxing stage of care. And so when I'm talking about parental behavior later, this is mostly what I'm referring to. And after this care is completed and parents have moved on, larvae then disperse out into the soil. And after about a month, they'll emerge to breed as adults themselves. So one question that should be occurring to you after seeing this reproductive cycle is, these beetles breed underground, under cover of forest canopy. So what kinds of temperatures are they actually likely to experience during parental care? And I actually tested this. I deployed a bunch of data loggers underground at my collection sites in Georgia for the duration of the summer breeding season. So in these plots, these dots, each dot represents a temperature reading collected during daylight hours in orange and nighttime hours in blue. And as you can see, even when we're taking into account the buffering effects of the forest, by the time we hit mid to late summer here, we're regularly seeing temperatures holding up around 24 to 25 degrees Celsius underground, which is well above that optimal 20 degrees. And so this field temperature data is what we use to calibrate our two treatment groups. In the laboratory, we set one incubator to fluctuate between 19 to 20 degrees, which, looking at this field temperature data, would represent a very benign environment. And this other one to fluctuate between 23 and 24 degrees Celsius, which really would be much more of a standard thermal environment, but for the purposes of comparison, we'll call this the harsh thermal environment. All right, so when we look at how our beetles performed in each of these treatments, we find about what you would expect based on previous data. So in our harsh thermal environment, warm thermal environment, we find that adult beetles that were continuously reproducing during their time in lab experienced shorter lifespans. And what's really interesting here is that regardless of the thermal regime, males in the lab lived about 50% longer than females. And so both of the sexes are facing this fitness cost of thermal stress, but for females, it's somewhat exacerbated because the reproductive costs for females are already elevated to begin with. And of course, it's not enough for our adults to suffer this survival costs. Larvae in the harsh environment don't do well either. Even when we were controlling for brood sizes, fewer larvae survived at the higher temperatures. And those larvae that did survive were much smaller in mass and not as healthy. But what I think is a lot more interesting to look at is how these larval fitness outcomes related to parental effort. So in this plot, you're looking at the relationship between standardized larval mass on the y-axis and standardized cumulative care on the x-axis, or the total amount of parenting experienced by larvae over a 30-minute observation, 24 hours post-hatching. So the three colors distinguish these artificially imposed social conditions in the lab, being uniparental male care in red, uniparental female care in gold, and then biparental care in blue. And so pretty intuitively, you can see biparental pairs tend to show the greatest cumulative parenting effort simply because there are two sources contributing. And so looking at this overall relationship in the benign environment, we find that increasing parenting effort is showing pretty negligible returns on offspring performance. But when we look at the same relationship in the harsh environment, there's a really interesting pattern that emerges, which is that instead of seeing an upward inflection of the curve reflecting more pronounced benefits of additive care, what we actually see is this weird negative parabolic relationship, meaning that offspring experiencing more care in this environment are more likely to be hurt than helped, which I find totally fascinating. So then the question arises, if care is more costly for parents in the harsh environment, and more care actually becomes detrimental to larvae under these conditions and doesn't help offset costs, do parents adjust behaviorally for this? And are adjustments sex specific? Because we ran our experiment as a repeated measures design, we can actually look at how the behavior of individual beetles changes from being in a uniparental to a biparental condition. So each line in these box plots connects measurements for an individual beetle under different social conditions. And so in this first example here, we see that females in the harsher environment don't attend larvae for quite as long, probably because larvae in this environment are developing, have shorter development times. But in neither environment do we see females consistently adjusting their duration of care based on the presence or absence of males. Whereas we can see males in both environments generally tend to abandon sooner if a female is present. Similarly, if we look at these other behaviors, so time spent regurgitating and time spent in carrion maintenance, we see that females are inflexible. They basically do the same thing regardless of environment, regardless of social condition. Whereas males do adjust their behavior, specifically by increasing feeding efforts in the absence of females, but only in this harsher environment. And so here we're actually seeing an environment by social interaction driving male behavior. And so where does that leave us? Do parents adjust parental behavior based on temperature-dependent costs and benefits of care? Males do, females do not. So very much in line with our understanding of sexual division of labor in this group. Even among the biparental species, males really tend to defer to females for much of the caregiving, particularly for the feeding of larvae, which I'm sure you're going to be hearing more about from Nick Royal, who's giving a talk later on in the series. But for here and now, what we've shown is that in addition to differing in social plasticity, the sexes also differ in their sensitivity to abiotic variables like temperature. which in turn means that the success of different care structures is going to depend on the environment, just not necessarily in the predicted directions. Okay, so moving on in my final minutes to some future directions. All this behavioral plasticity data is obviously super fun and interesting, but how do we ensure that this kind of empirical data we're generating can be used to better understand how organisms are responding to climate change? And for me, there's really two components to this. The first is to get a better handle on how and why individuals in social groups or in populations vary in their plasticity. So taking sex as an example, if males and females we consider to be distinct units of the population that are going to show distinct tolerances and preferences as they move through reproductive stages, then models calibrated from the thermal responses of a single sex are not likely to give us a full picture of how a population is experiencing climatic changes. And by assuming that the two sexes respond similarly, we could be missing some really important changes both in reproductive output and in patterns of selection on thermally adaptive traits that may be driven disproportionately by one sex. And a current limiting factor here is that we just don't have a very strong framework to make predictions about how the sexes will differ in something like thermal plasticity. And if there are differences, what are the mechanisms underlying those differences, right? So are they driven by physiology, by behavior, or all of the above? So one research effort we're currently undertaking in my lab, my new lab at VT, is we're trying to take this question apart piece by piece and actually test specific predictions about how males and females should differ in these traits, given what we know about their divergent reproductive strategies. So right now we're using this model of plethodontid salamanders, which are these montane-adapted semi-fossorial amphibians that are highly attuned to climatic cues to drive their patterns of surface activity. But another really interesting thing about them is that reproduction is more or less a constant factor affecting their physiology and behavior. Because for ectotherms inhabiting cold environments, a single growing season is often not long enough or warm enough to support a full reproductive cycle, these guys really have to draw it out. So it takes females almost a year to yolk a clutch of eggs, and males really have to be ready to mate at any point during that window. And in all this time, they're experiencing changing seasons and constantly need to acclimate their physiology and behavior to maximize returns. So one way that we're looking at this is we're trying to characterize sex-specific thresholds for surface activity in the laboratory, predicting that, as we observed in my skinks, females might show a more conservative strategy when acclimating to changing thermal regimes. Another approach we're taking simultaneously is using the technique of respirometry to characterize changes in male and female metabolic rate under varying thermal acclimation treatments to understand the extent to which sex-specific activity patterns may be baked into their physiological responses. And something that I think is going to be really powerful here is carrying out these experiments in conjunction with each other to really understand how these mechanisms operating within individuals are related to each other, or whether the covariances themselves might actually differ, show different relationships between the sexes. A second component of this that I've become quite fascinated with over the years is the extent to which the environment shapes the social interaction itself. So looking beyond just whether individual participants in an interaction modify their behavior in response to an environmental change, are we seeing that the actual impact of one individual on another changes depending on environment? So here we can borrow the concept of the interaction coefficient from indirect genetic effects, which is a mathematical framework for describing changes in phenotype that result from changes from interactions within the social environment. And to give an idea of what this might look like, we can consider a scenario of family dynamics where one caretaker's effort is contingent upon another. So there's a social effect on parental behavior. But let's say that the magnitude of that effect is going to depend on the prevailing environment. So even if the input from the social environment is remaining constant, psi, or the interaction coefficient, is not. Simultaneously, in this same scenario, we could be considering parental effects on offspring. So perhaps under benign conditions, the amount of care received has no additive benefits for hatchlings above a certain threshold, like we saw in my beetles, but put the family in a new and harsh environment, and suddenly hatchlings show very distinct responses to different levels of care. So now we have an environmentally dependent psi describing effects of parental care on offspring. What I'm describing here is not anything special or groundbreaking. We see this kind of behavioral plasticity everywhere we look in the natural world. But what I think could be really interesting and exciting about reframing these patterns in an IgE framework is that it gives us a method to leverage empirical data to parameterize models that actually test how social behaviors could be evolving under different climate change scenarios. Okay. So I want to end on that note to leave time for discussion and for other folks to weigh in. I know I've just spent a lot of time talking about reptiles and insects, and there's a lot of bird and mammal folks in the audience, so I appreciate you bearing with me, and hopefully some of these ideas resonated with you. Before opening up for questions, I just want to acknowledge my funding sources, the universities and the lab groups that hosted this work. And most importantly, all of my mentors and colleagues and everyone who contributed to making this work possible. And with that, I'm going to try to bring up my faces again and open it up for questions.
Thank you, Jen. That was really an exciting and fascinating talk. I'm sure we have lots of questions from people. So at this point, I'm just going to open up the floor for questions. Just a reminder, when you have a question, please type a question mark in the chat and I will call on you and then you can ask the question to Jen. If you're on YouTube, please post your actual question in the chat on YouTube, and I think Adriana's monitoring that and will let me know. So at this point, and by the way, when you ask a question, again, we're trying to build community, so please tell us your name and kind of what your general research interests are. So at this time, please, if you got questions, put them in the chat, and looks like we already have one from YouTube. So Adriana, why don't you start us off? Thanks.
Hi, so this question is from Rachel Schultz, and it said, do ectotherms tend to display more behavioral plasticity in response to thermal environment than endotherms?
Yeah, that's a really good question, and one that I kind of grappled with a little bit when we were writing that review. And I'd say that it's not necessarily more or less behavioral plasticity because both endotherms and ectotherms are going to behaviorally thermoregulate. I'd say it's more in sort of the relationship between ambient temperature and body temperature. And so in ectotherms, we have ambient temperature exerting this direct impact on physiology, whereas in endotherms, we have really, it's more about the differential between ambient temperature and body temperature that's going to determine how much energy is required to sort of maintain homeostasis. And so I wouldn't say that there's necessarily a hard and fast ectotherms are going to show more or less behavioral plasticity in response to temperature. It's more that the mechanisms by which endotherms and ectotherms are going to adjust their physiology, which of course underlies behavior, to accommodate different thermal environments is going to be distinct. And so we just have to think about, depending on our system and where it's located and what kind of thermal variation it experiences, what are those mechanisms that are available to that species?
Okay, thank you for that. And I'll go to the next question. Carson, I'm going to skip down to David and then come back to you, if you don't mind. So David Fisher, please go ahead.
Oh, hi. Yeah, my name is David Fisher. I'm a lecturer at the University of Aberdeen in Scotland.
Hi, David.
Nice to meet you virtually, Jen. Been
Yeah.
reading your stuff for a while. So I thought all of that was really, really fascinating. I guess what I was wondering was, you talk quite a lot about selection on males and females, and I wondered if you had any thoughts about sex-specific heritability or cross-sex correlations in these traits. Like, if these traits are really tightly genetically correlated, then it doesn't matter what selection's doing; they aren't going to be different. So, do you have any estimates of repeatability or other things that might give us an idea about what's going on in terms of genetic variation, sex-specific genetic variation?
Yeah, for sure. And I will be totally honest and say I don't know about heritability. I think that's that opens up a whole nother additional can of worms, is thinking about if there are these differences in behavioral traits between the sexes, are they more or less heritable from one sex or the other to individuals' offspring. And yeah, not my area of expertise, but I think that's a really fascinating additional avenue for future research.
Okay. So I'll go next with Karsten.
Yes, hello, my name is Carsten Schradin from the University in Strasbourg, France, currently in South Africa doing field work on mammalian social evolution. Thanks a lot for fascinating talk, Jen, with different aspects and different study systems on the effects of thermal harshness. I have a couple of questions to the burying beetles to understand it better. I was surprised that 20 degrees is benign for them and 24 is good. You said quickly that this is kind of known, but I missed it. Can you repeat why a priori you expect the 24 to be a bad temperature? And the second part to this, the second question to this part is, you showed fitness differences of the larvae in number of larvae dispersed out of 10 and mean larval mass, but do you also have measures on total fitness? I mean, total number of larvae raised till dispersal? Or... Yeah, these two questions.
Okay, sure. So first question, why did we expect a priori that this 25-degree temperature was going to be harsh? So there'd actually been a number of laboratory experiments preceding mine that maintained beetles at these temperatures. And again, like similar to my thinking, when I deployed temperature loggers at my site, I really expected them to handle the higher temperatures better. But they just don't. There's just a lot of high survival costs that occur at those warmer temperatures. So those previous studies, actually, I used the two different thermal extremes, whereas the previous studies actually used the gradient of temperatures to show that reproductive fitness kind of peaks right around 20 degrees and then declines at temperatures above 20 degrees. So there'd been this a priori research in the lab. There'd also been this previous research in the field showing that beetles that are found breeding at warmer temperatures, they're dispersing fewer larvae, just not producing as healthy of larvae as well. And so, yeah, I mean, I think there was plenty of evidence there that kind of set up for those temperature differentials. And then your second question was about the number of dispersing larvae.
Yes, the total. I mean, your graph shows out of 10, but if in the hot environment they produce double as many larvae.
Yeah, so that was a decision that we made about the experimental design, because These beetles will actually, they have mechanisms for brood reduction where they'll actually cannibalize a lot of their larvae, and that can account for a lot for large differences in brood size. And they can also have different numbers of eggs to begin with. And we were really trying to get at whether if we saw differences in larval number, how was parental behavior specifically affecting that? And so we controlled brood size for all of our families by essentially collecting all the eggs that were laid and then introducing 10 larvae to each of our brood balls. So they all started with the same number of larvae, which allowed us to determine how much the reduction in larvae was attributed to the effort of those parents, as opposed to necessarily just the starting number of larvae that colonize the brood ball.
Okay, thank you.
Thanks.
If other people have questions, please post them in the chat. I'll go to you ahead of me. I see Adriana, so why don't you go ahead? No? Steve, I'll go with Steve Trumbo, please.
Hi, Jen. Thanks for a great talk.
Hi, Steve.
Enjoyed
Great to meet you
hearing
virtually.
that. So I had a question about burying beetles too, with the, that paradoxical result you had with more care in the, I believe it was in the harsh environment. Actually, the larvae did worse. I didn't catch all the details. It was like a 30-minute observation you did.
Yeah.
And what were you observing, or what behaviors were you recording?
Yeah, so this was a 30-minute scan sampling survey of parents 24 hours after larvae colonize the brood ball. So sort of hitting them during when we estimate is the peak care, and using that as a representation. We were recording instances of what we call direct care, so parents feeding from the carcass and provisioning larvae, versus instances of indirect care, which is we classified as parents maintaining the brood ball, moving about and secreting on the brood ball. And then we also recorded instances of no care, so they went off and buried themselves in the dirt. And so that graph you were seeing of the negative parabolic relationship, that was where we combined. So we pooled all of the instances of indirect care and direct care and took that as a cumulative measure of care experienced by offspring.
Yeah, I'm sure you've thought about how using temperature with an ectotherm as your harsh environment, it affects, of course, many other things. At the warmer temperature, they might be at a slightly different stage of development than at the cooler temperature. The parents may have to be more active or feeding more. I was wondering if some of those behaviors are actually the parents' self-maintenance rather than giving care, like feeding from the carcass. Could you tell when they're using it for self-maintenance versus getting ready to regurgitate to the larvae?
Yeah, that's a really good question. So typically within our surveys, what we would observe and why we kind of ended up pairing the feeding from the carcass with regurgitating to larvae is that typically those behaviors would occur in sequence with each other. So parents would feed and then regurgitate. Whereas a lot of instances where we observed parents not regurgitating to larvae at all, they weren't necessarily spending a ton of time on the carcass feeding themselves. In the instances where they just weren't being parental, they would kind of just go off and bury themselves in the dirt and just check out entirely.
Right. The last question, what was the size of the carcass?
This was, if I'm remembering correctly, this was like a couple years ago. We used a 30-gram carcass, I believe. So we actually set it on the larger side of what we would typically use for these experiments to account for the fact that at the warmer temperature, these carcasses would tend to have shorter lifespans. if you will.
Yeah, so with the having a limit of 10 larvae to care for, which they normally would raise more than that on a 30-gram carcass, I mean, that might explain one reason you didn't get this big effect on more care leading to more success, because there were plenty of resources for everyone.
Yeah, which is why I was really surprised that we still had fewer larvae surviving in this harsher treatment. You know, they had plenty of resources, and so we were really trying to whittle it down to you know, if there are differences emerging between the clutches, like what is it about parental behavior that is contributing to that.
Thanks. Yeah.
Thank you so much.
I have a couple of questions. I can interdisperse them through as people put more questions in the chat. I'm going to come back to that, if you don't mind, the work where you were, I think it's it's a lizard or skink, I apologize. You're looking at daily activity scores. And I think this kind of comes back to Steve's question a little bit there too. In that daily activity scores, what behaviors, and I apologize if I missed this, what behaviors were most important? Were they spending more time defending, competing for mates, or was there more time foraging? How did that play a role into that activity score?
I wish I could answer that question more with like what they were doing. So we were out observing these skinks pretty much around the clock every single day. And while they lend themselves quite well to detection and basically ability to chart their movements observationally, most of their actual behaviors, including, you know, foraging and social encounters and copulations, are pretty cryptic. And so we recorded a few very rare instances of actually seeing individuals attacking each other or mating with each other. But for the most part, we were working with spatial position in the enclosure. And so when we generated that activity score, it was constructed entirely from did we see the lizard out and about at all? If we saw it, how many times did it move? I think it was how many times did it move further than a meter? How many times did it move between these pallets that we were using as territories and defensible resources? How many times did it move between different sides of the enclosure? So it was a bunch of these different scores, all related to how much they were out and moving within the enclosures.
Okay, sounds like Daegu problem. Same problem I have. One more question, if you don't mind. So again, looking at that figure, I've been thinking a lot about inter-individual variation, you know, and even though the slopes for the males, the reaction norms or slopes are going up, you know, in terms of activity scores and daily temperature, do you see any mechanism or explanation for why males vary so much within treatment? So like, for example, with the Daegus, we've been exploring the importance of masculinization and how that affects communal care and fitness. Are there any other mechanisms that you're exploring in that regard?
Yeah, so how much males vary within a thermal treatment? So within the skinks, I would think that a large contributor could be body size and dominance to an extent. That's just my first instinct. Again, we weren't necessarily able to characterize or directly observe a lot of the aggressive or mating male-female interactions, but I suspect that there could be some variation attributed to sort of position within the social network and how they're excluding each other from different areas that could somewhat influence the movement and the space use itself. But as for, so obviously I've moved on from the skinks, and now my current work is looking more at these salamanders. And so, yeah, I think a lot of the mechanisms that we're going to be really interested in investigating for accounting for this individual variation. Yeah, I mean, we're really thinking a lot about individual variation between the sexes, but I'm really curious to think more about individual variation within the sexes as well. Yeah, I mean, what are your thoughts on that, given your experience with degus?
Well, we see a lot of variation due to the masculinization effect of intrauterine position. But I was wondering with these guys, do they have like throat pouches or anything, like the color pouches that you can manipulate to see if, dominant status influences the difference in how they respond to the environment?
Yeah. Yeah, I see. No, that's a really good question. Yeah, these guys, the skinks are actually pretty monomorphic.
Okay.
They don't have, the males don't have any huge ornaments or anything terribly sexually dimorphic about that, which is why I think they made a fairly interesting system for investigating these questions, because a lot of the behavioral differences are really just down to different reproductive strategies, more so than hugely divergent, ornamentation or anything like that, sexually dimorphic features. But yeah, I think that's a really good point. That's going to be really relevant for a lot of other systems.
I'll probably talk more with you later. I think
Sure.
some of Steve's ideas about developmental stage too are important.
Yeah.
I want to give other people a chance to ask you questions now. But let's come back to that. That's really cool. I think Adriana was next.
Yeah, I didn't want to take over your turn, Lauren. Okay. Jen, thanks for your talk. It was really, really cool. I want to go back to your mesocosm experiment, the shade and the non-shade. I mean, I don't know if you were expecting those results, where the warm environment you had less interactions, and then in the cooler environment you had more interactions. I don't know if that was your expectation for the experiment. I think if I were to run it, that would have been mine somehow. But I'm wondering if you have any insights on the benefits of these interactions in your specific study system. And a second question that probably goes before that is, what's the normal temperature for them? Is it the shade or the sun more like to be? Yeah, those are my two questions here.
Yeah, no, thank you. So what that result you're referring to with the greater in the greater interactions in the warm environment. So I think that came from that last slide I showed, which was Alex's follow-up study, where she ran the same experiment in the summer. And so that was like in peak summer temperatures, we get this reverse effect of reduced activity and reduced social cohesiveness in the warmer enclosures, which is opposite to what I found in the spring when temperatures are cooler. So basically we see this shift happening from sun or from cooler temperatures being really restrictive to now cooler temperatures once you hit the hot summer period being more alleviating, and individuals are able to get out and interact a lot more. So to circle back to your specific question about that, can you remind me one more time?
Yeah, no, if you have or explored the associated benefits of your interactions. Because one of your statements basically it was kind of going into the direction of, now that we have this difference or effect of temperature, then we might be affecting, there's an effect through the social behavior, right? So I was wondering, to have that effect, we should know a bit more about what benefits do these social interactions have. And so I was wondering.
Yeah. So the social structure of these skinks is largely centered around this family unit. And so they're considered socially monogamous, where males and females establish and defend a territory together. And then the offspring reap the benefits of that monogamous relationship by also being tolerated to remain in the territory with their parents. And so when we talk about social interactions that are beneficial to this species, we're largely thinking about interactions within this family unit during the period when offspring are present, so sort of summer onwards, which is why I showed that data slide of Alex's study, looking at the extent of social interactions and how that related to temperature. But going back to the breeding season, I think, of course, we see a different benefit to different kinds of social interactions, where males and females, while yes, are socially monogamous, like in many species that are socially monogamous, of course, mate outside of the pair and gain reproductive advantages by interacting with males and females that are outside of that that relationship. And so I think we see that clearly with the males who move around the most, having the highest number of mating successes. Though interestingly, the individuals who have the most opposite sex encounters also have the most same-sex encounters, which tend to be more aggressive. Again, this is all based on spatial positioning, not based on actual observation of interactions, but we can infer from those opportunities for interaction that the more connected individuals during the breeding season are both going to have more opportunities to mate with opposite sex individuals and more opportunities to potentially be having negative interactions with same-sex individuals that could be establishing, dominance hierarchies of some form.
Cool system, thank you. And I forgot, Adriana Maldonado Chaparro from Universidad del Rosario in Colombia, and I study social behavior, particularly social structure.
Yeah, I'd love to chat with you at some point about social network analysis and
awesome, sure.
Yeah.
Next goes to Karsten.
Yes, I have another question, and I think it's very similar to what Lauren asked and what has been the topic here for many of your answers in your talk, but I'm a slow learner, so I still ask it again. The difference between the males and females, I mean, you showed in your talk the nice photo of these the brown beetle species with the very big males and small females, but your two study species, the skinks and the burrowing beetles, I think there's no real sexual size dimorphism, meaning the differences between the sexes is mainly explained by their reproductive tactics, the male lizards being more successful when they're more active, so they meet more females, and the male burrowing beetles, when the female is around, leaving earlier, probably also looking for other females. Is that correct? And if so, wouldn't it best your next study species to be one with real big sexual size dimorphism to address the question you have been asking here all the time, that there should be a bigger difference between the sexes when they also differ more in body mass?
Yes. For sure. I mean, I do think that there is a lot of interesting dynamics to explore in more sexually monomorphic species when related to these questions, because of the fact that, if there are really large differences, we kind of can attribute those mostly to reproductive tactic. We sort of have a direct way of discerning that by minimizing huge differences in dimorphism between the sexes. But to your point, that shouldn't I be really wanting to exploit more sexually dimorphic species to set up the precedent for the sexual dimorphism and thermal plasticity? Agreed. And so my current system, plethodontid salamanders, is really interesting in that respect, and because they are sexually dimorphic, females tend to be larger than males. But more interestingly, we also have, where I'm located, we have an elevation gradient. And so what's interesting about the elevation gradient in salamanders is we have this grade of season length, and at the higher elevations, we actually see a more exaggerated degree of sexual dimorphism, because females really need to be as big and have really large fat stores to be able to reproduce at these colder, higher elevations. And so at lower elevations, we see less pronounced sexual dimorphism. And so not only do we have that sexual dimorphism in our main study population, but we also have this comparison population. And so we can actually try to start teasing apart whether, like, getting at the predictions of this. So our species and systems where we see a greater degree of sexual dimorphism, do we then actually expect to see a greater degree of sex difference in the thermal responsiveness as well? So that's something that I'm definitely hoping to tease apart more with my future work.
Okay, thank you.
There's just a few of us left. If anyone wants to ask questions, just jump right in. I can also stop the stream to YouTube. Get a more relaxed conversation. Do that. I don't see any more questions in the chat. But with that, I'm going to stop the YouTube stream. And before I do that, Jen, I want to thank you publicly. That was fantastic. Really cool stuff.
Thank you. Thank you so much. Super fun. And yeah, really, really great group to be chatting about these topics with. So thank you for hosting me.